as you watch this video, there are thousands, I'm sorry, billions of people on this planet that are sitting about doing their daily tasks. Some of us eating, sleeping, doing what God knows what. But the point is, our bodies are super complicated, and it's running through a series of very complex mechanisms. And these mechanisms are really directed through the control of the cells in on a molecular scale, and that is DNA. And if I were to ask a little kid over here, and I ask him, hey, little boy, what's the organ in our bodies that's responsible for controlling everything? The control center, he's going to say the brain. And if I were to shrink down on a metabolic scale and ask the cell the same very question, the cell will be answering the question by saying it's the nucleus. And the nucleus is where the DNA is stored. So let's talk about DNA replication. When these cells are dividing, they have to divide everything inside them, essentially, including mitochondria, cytoplasmic, whatever the hell's inside them. Everything has to be duplicated before the parental strand can be divided into two identical daughter strands, or I'm sorry, two daughter cells. And before this happens, DNA has to be replicated. DNA replication happens in the S phase. The S phase is when let me just highlight this for you. DNA replication occurs during the S phase. Before we discuss the exact mechanisms of DNA replication, let's think about what the people were thinking before this was made an established fact. So there were a couple theories that suggested how DNA replicates, and the first was called the conservative replication. The conservative that was me running out of breath. So conservative replication happens when we have a double-strand DNA going into another double-strand DNA, but at the same time, it's copying an entire other double strand somehow. This is called a conservative replication. And the other possible suggestion was the dispersive replication. Dispersive replication happens when we have double-stranded DNA that's kind of like is replicating, but then the offsprings are going to have a mix of replicated and the original DNA, and that's represented by the blue and the red. The red being the newly synthesized DNA, and the blue being the previously synthesized DNA. And then the other suggestion was that DNA is semi-conserved. And this is exactly how DNA replicates. And just to represent this by stick figures real fast, if we have a double-strand DNA, blue and blue, one of these strands is going to go its own way, and the other is going to go its own way. And because they're semi-conserved, they will produce the same exact DNA, double strands. And this is exactly what's happening. And here's another example of the same thing. Don't focus on the nucleotides. It's just an example. They're just there for God knows what reason, just to show you that there are nucleotides there. But look at the color. So the blue represents the previous parental DNA strand, and it goes into its own directions. And the red and the green, they represent the newly synthesized DNA. And they both are the exact same thing because they're complementary to one another. But, sometimes we have mutations and they can cause slight differences. And that's important to talk about later on. So here's a quick recap of how DNA is wrapped around histines. So DNA is linear and it's wrapped around histines. 8 histines are called 8 histine molecules come together to call something called nucleosomes, and we've talked about that before. So before DNA can be replicated, it's very coiled up. It has to be uncoiled. It's uncoiled by an enzyme called the helicase. So the helicase is breaking up the strands. Basically, it's allowing the DNA to form this very shape that allows other enzymes to come in and allow replication. Before we discuss that, here's a question to ask. How do you think the helicase knows where to stop and start unbinding? Because, look, we have we have a lot of DNA. So how does the DNA know where to stop and start replicating? Well, the start codons are areas which are specific nucleotides that indicate the helicase to stop and start from unbinding from that point. And that point is called the point of origin, the origin of replication. In eukaryotic cells, we have 3 different proteins that locate the start codon. And they're assisting the helicase with that. Whereas inside prokaryotes, we just have one cell that does the same job. It's called a DNA. And here's a very interesting, well, a very common, realization that eukaryotes have a more sophisticated response in comparison to prokaryotes. Now, imagine that there were two ropes, and these ropes are tangled up together. This one rope is tangled up together, and you were trying to pull them apart. And as these ropes are kind of being pulled apart, yes, the front is becoming more freely open, but the upstream of the ropes are kind of like getting more stressed up and more tensed. And the exact same thing happens with DNA. And there is another set of enzymes that help, release that tension. And this tension is released by cutting the DNA. And this is done by cutting the DNA. And we're going to talk about that in a second. So the enzyme that allows this extra stress being created because of the helicase unwinding is released or reduced by enzymes called the topoisomerases. I hope I said that right, but there are two kinds of topoisomerases. The topoisomerase 1 and the 2. The 1 uncoils the one DNA strand. So there are some parts of the DNA that are now singular because of replication. And the tension over there is released by the topoisomerase 1. Whereas if it's a double strand, the uncoiling happens through a topoisomerase 2. And remember, it's all upstream. And you might be confused what upstream and downstream is, and it's really just a reference to the point of origin. So if this were the point of origin, everything that's upstream is called upstream, and it's indicated by the negative sign. Whereas if it's downstream, it's going from the point of origin, it's called and it's indicated with the plus sign. We're going to talk more about this in future videos. It's going to make more sense. But as of now, we can just get the overall concept. So here's the exact mechanism of DNA replication. So when the helicase is, it's going off in opposite directions, and remember the helicase is always going from the 5' to the 5' to the 3', just like DNA replication. And when that's happening, we have the point of origins, we have the point of origin being formed in two sides. One of them is going this way, the other is going that way. So once the helicase is able to unwind the DNA, these nucleotides are exposed. And we know that nucleotides are reactive because we have talked about this before. The structure of the nucleotides can form bonds with surrounding molecules, and we don't want that to happen. So we have something called the single-stranded binding proteins that will come and bind to the nucleotides temporarily while it's being unpackaged in preparation for replication, and it really helps the entire process. And this whole complex right here is known as the open complex. Okay, now we have the open complex ready, so we can start. So we have something called the DNA polymerase, which will then bind to the strands and then allows the replication to begin. So this DNA strand right here, the DNA polymerase can start by binding only. So if we have a DNA polymerase, right, it's just trying to attach. It has to attach through a RNA primer. So an RNA primer is synthesized, has to be synthesized for each template for DNA replication to begin. So when we have RNA primers, they come and attach to the template strands. This allows the DNA polymerase to attach and begin replication. And when that happens, this is assuming that, this is not, let's assume that we have the RNA primers attached on each of the templates. Then we can have RNA polymerase going in the 5' to 3 direction. So that's happening. And when that happens, we have replication that's happening. So once the entire DNA is replicated, we have to replace the RNA primer at the end as well. And that's made possible through something called the ligase. The ligase will replace the primer with DNA, because remember, it might be RNA primer, which definitely allows DNA replication to begin, but it has to be replaced at the end with real DNA. That's important, because remember that DNA, for example, cannot have uracil. It cannot have uracil, and an RNA primer might have uracil, and it has to be replaced therefore. Well, we have said this before, that eukaryotes have a more complicated mechanism of replicating, and this involves the uncoiling of histines. And this should be made obvious with this diagram, that these linear DNAs are wrapped around histines, and these, if it's being translated, if it's being duplicated, then it has to be uncoiled from the histines.